This mini lecture is on the isolation of bacteria colonies using the streak plate technique, and you can reference Chapter 3.4 for more information in the Foundations of Microbiology textbook. The objective for this lecture is to be able to explain how the streak plate technique results in isolation of a single colony from a single seed microbe. Here are the key concepts in the lecture, and basically what you should take from this is that one colony that is isolated using the streak plate technique is equivalent to one species which is under examination, and more importantly is that using the streak plate method, it is important to remember that bacteria grow in colonies when given adequate space and nutrients, and when they are grown on a medium with the nutrients in a favorable environment with proper conditions that will promote their growth. To begin the isolation technique, the supplies you're going to need are a media that is going to support the growth of the bacteria that you are examining, and it can be a selective media. It can inhibit the growth of the bacteria that you don't want to examine. It can be differential in determining which of the two bacteria are in the culture. It can be enriched and promote the growth of all different kinds of bacteria. And it's going to be on the agar plate. And then you're going to need a cell culture. And more importantly, that whatever media you choose, it's going to have nutrients that promote the growth of the bacteria under examination. When culturing for isolation of a bacteria culture, it is much like seeding microbes in a different type of soil depending on which media you choose. And in a bacteria culture that could be mixed or pure, it can have a variety of different microbes. And by inoculating a media, it is much like planting the seeds in different types of soil. And depending on which type of soil you choose, the seeds will grow differently. And see, I've drawn here that seeding the microbes in the soils, it could be a selective, differential, or enriched. To perform a bacterial culture, the first step is to inoculate a media using the streak plate technique, which I will demonstrate to you. So here is your culture, and what you're going to need is the media that you choose that's going to help identify the bacteria under examination. And you're going to have an inoculating loop, which I will use as this marker. So you're going to dip it into the culture, and you're going to take, you're going to basically streak this plate in the first quadrant. And right here it's going to be very thick, and the bacteria are going to be overlapping each other. And then you continue to inoculate the loop and streak the plate in the different quadrants. So see, I swiped through a corner of this to help spread out the bacteria more. And then I'm going to take a little bit from of the bacteria over here. I'm going to streak a little bit more, and see the lines are getting spread further apart. And then finally, I just kind of do a swipe here and here, and then the bacteria should be spread thinly in this region. Microscopically, what you're going to find is that in this quadrant, we'll look under the microscope figuratively, that the bacteria are all clustered. They're overlapping each other, and when you incubate it and they grow, you're not going to be able to find colonies because they're unhappy, they're overcrowded. Then in this quadrant, where the bacteria spread very thinly, you're going to find the bacteria are spread apart. After incubating the petri dish and allowing the proper time and temperature and atmosphere conditions for this bacteria to grow, you'll eventually find the isolated colonies, in the third and fourth quadrants, which will be right here. And as you can see that these bacteria grew and they're very crowded and overlapping, but as you will see that as it's spreading out, you'll be able to find that single bacteria were spread into this region and they were happy and they were able to grow into large mounds rather than these smaller dots. But this is how you find the isolated colonies, and from there you would take samples from these colonies and you'd be able to put it on a plate and view it under the microscope to identify which microbe is growing best on this plate.